Good day, this is Professor Will, CBMD PhD. Today's Sunday, August 25th, 2019. It is 23:40 and 20 seconds Eastern Standard Time, and this is a short research study on DNA sequencing. So let's get started here. Advances in DNA sequencing. Computing automation technologies have allowed the sequencing of entire genomes from living organisms. Bacterial genomes range in size from 0.5 to at least 10 MB, 1 times 10 to the power of 6 base pairs, and are composed of approximately 90% coding regions. Therefore, high-throughput sequencing of randomly cloned DNA fragments represents the most cost-effective way to rapidly obtain genome information for these organisms. Since the publication in 1995 of the first whole genome sequence for a bacterium, over 60 microbial genome sequences have been published, and high-coverage sequence data have been added to the public domain throughout this period for many other genomes. Sequencing of a microbial genome requires logistical planning for a challenge of high-throughput sequencing, bioinformatic analysis, and presentation of the data in a concise, user-friendly format, usually in the form of a World Wide Web site to supplement a journal publication. This is an intensive process that is often underappreciated. Following the sequencing analysis and initial assignment of function based on similarity to previously identified genes in sequence databases, analysis of gene function at the laboratory bench is required to confirm gene function. These functional genomic studies will also require substantial scale-up in order to keep up with the high-throughput pace of genome sequencing. Simply stated, in the pre-genomic era, genes were usually identified based on the selection for function, followed by the laborious task of cloning, subcloning, and sequencing. Genome sequencing has reversed this process today. And the challenge of the post-genomic era will be to determine how all the gene products of a genome function, interact, and allow the organism to live. Introduction to DNA sequencing. DNA sequencing has seen many technological changes during the past 2 decades, evolving from slab polyacrylamide gels that could resolve 20-50 BP at a time to the 700 BP reads of today. In the late 1970s, DNA sequences were manually read from X-ray film using chemical and enzymatic methods to incorporate radiolabel. During the 1990s, fluorescently labeled DNA was resolved on slab gels and read using computerized base calling programs using sequencers such as the Applied Biosystems 377 model. In the late 1990s, capillary sequencers such as the Emerson Megabase and Applied Biosystems 3700 models were introduced and dramatically increased the throughput of automated DNA sequencing. The availability of these instruments was the catalyst that allowed the shotgun sequencing of the fruit fly genome and greatly accelerated the schedule for completion of the Human Genome Project. Upon publication of the 1.8 MB chromosome of Haemophilus influenza in 1995, it became apparent to the scientific community that sequencing of bacterial and even eukaryote genomes was possible by scaling up existing sequencing technology. Although the utility of the whole genome shotgun approach for sequencing microbial genomes is unquestionable today, the original H. influenza project was in fact deemed an unproven high-risk venture. It was not funded because such a large segment of DNA had not been sequenced by a random approach. The standard paradigm for any large-scale sequencing projects was based on strategies used for sequencing the human genome and related model organisms. These top-down techniques consisted of 2 phases, an upfront mapping and cosmate ordering phase, followed by subcloning and sequencing of a minimal tiling path, a complete set of cloned genome fragments with minimal overlap among the cosmates. Sequencing of larger segments of a microbial genome was not attempted due to a computational limitation. At that time, existing software packages for the assembly of random DNA fragments were not sufficiently robust to assemble a DNA segment much larger than the size of a cosmate insert, 40 kbp. However, advances based on an unconventional approach to sequencing the human genome set the stage for whole genome sequencing of smaller genomes. The Venter Laboratory at the National Institutes of Health, NIH, and later at the Institute of Genomic Research, TIGR, developed the Express Sequence Tag, EST, method for streamlining gene discovery and human genome sequencing by enriching for and sequencing messenger RNAs. By optimizing the conditions for high-throughput fluorescent sequencing and developing the computational software to assemble hundreds of thousands of random sequences into contiguous sequences, contigs, the stage was set to attempt a shotgun approach to sequencing an entire genome, which had last been done for bacteriophage gamma in 1977. Microbial Genome Sequencing Project, Library Construction Phase. Library construction is the most critical phase of a microbial genome sequencing project. Assuming a completely random library, Lander and Waterman calculated that the sequencing statistics would follow a Poisson distributed, i.e. sequencing enough random reads to add up to one genome equivalent would leave 37% of the genome unsequenced. Sequencing of non-random library will result in a significant deviation from the Lander-Waterman model for random sequencing, and the generated assemblies will have too many gaps to be efficiently closed. A library construction procedure has been developed. Fleshman et al. 1995 to achieve a high order of randomness, and it consists of the following steps. Number 1. Random shearing of genomic DNA and purification of 2 kbp fragments. Number 2. Fragment and repair and ligation to blunt-ended dephosphorylated vector. Number 3. Gel purification of the V plus I vector plus single insert banned from other forms. Number 4. Polishing of free ends and intramolecular ligation of V plus I DNA. Number 5. High efficiency transformation of Escherichia coli strains DH10B, Schur, etc. Number 6. Direct plating onto 2-layer antibiotic diffusion plates. The optimized method results in efficient sequencing because the library is a collection of highly random single insert fragments that can be sequenced from both ends using universal primer. Key factors for success include random shearing of the DNA, usually by nebulization purification of a narrow size range of insert using minimal amounts of long-wave UV light. A second, gel purification of V plus I to minimize plasmid clones lacking insert or containing chimeric inserts, propagation of clones in a highly restriction-deficient E. coli background, and outgrowth of transformed cells as individual colony-forming units on an antibiotic diffusion plate rather than standard outgrowth as a mixture of clones and liquid medium, which may select against certain slower-growing clones in the transformed population. Even with all these safeguards in place, however, gaps in the genome do occur. This is due to both the statistics of random sequencing and the inability to clone certain DNA fragments such as those containing strong promoters, e.g. the 16S rRNA promoter and H. influenza, or toxic genes e.g. the complete HIN3 restriction enzyme gene without the cognate methylase gene present in the transformed E. coli strain. Random sequencing phase. After the library of constructed random sequencing of the individual clone is performed, this involves 1. The production of tens of thousands of templates in 96 well blocks, 2. Sequencing of the purified plasmid template DNA in both directions, 3. Transfer of the edited sequence reads to a database, and 4. Assembly of the sequence data into contigs. Editing and annotation phase. Following genome closure, the sequence is edited and annotated. Editing consists of proofreading the sequence and resolving ambiguities or regions of low. Coverage by dye terminator sequencing. In addition, possible frame shifts are identified, PCR amplified from genomic DNA and resequenced. It should be noted, however, that some apparent base insertions, deletions, and frame shifts are authentic. Examples include polymorphisms and repetitive sequence of Campylobacter jejuni genome and more than 20 authentic frame shifts in protein coding regions in the lab-propagated H. influenza RD organism. Several of the counterparts of these genes and other H. influenza strains have been demonstrated to full-length functional genes. Annotation begins with gene finding, which is not an insignificant task in prokaryotes. Although stop sites of open reading frames ORFs are straightforward, identifying the proper translation initiation codon can be challenging. Annotation also involves characterization of other features of the genome, including repeat nucleotide sequences, gene families, and variations in nucleotide composition. Furthermore, all genes are classified into functional rate categories and analyzed as a whole for the presence of various metabolic pathways. In this way, whole genome sequencing provides a complete picture of the metabolic potential of an organism and lays the groundwork for many follow-up studies, once the genetic complement of the cell has been defined. The Bioinformatics Database. The Genome Project monitoring sites given are among the most comprehensive and are continually updated as new genome projects are both initiated and completed. In fact, it is estimated that in addition to the 60-plus microbial genomes completed 7 years after the publication of Haemophilus, the Genome Project websites listed provide extensive annotation role categorization search capabilities and genome segment display and retrieval. In the near future, experimental data from functional genomics work expression of gene under various conditions, phenotype, of knockout mutant protein, protein interaction information, etc. will be incorporated into these existing structures and provide much added value to the basic research community. Bioinformatics, biological informatics, specifically the computer-assisted analysis of genome sequence and experimental data. DNA microarrays, miniaturized arrays containing thousands of DNA fragments representing genes in an area of a few square centimeters. These chips are used mostly to monitor the expression of genomes at the level of messenger RNA. Functional genomics, the study of gene function at the genome level. Genomics, the study of the genome, or all the genes in an organism. Proteomics, the study of the protein complement, or proteome, of an organism. Whole genome shotgun sequencing, a random approach to genome sequencing based on shearing of genomic DNA, followed by cloning sequencing and assembly of the entire genome, and ultimately leading to finished annotated genomic sequence. Sequencing of entire genomes can also be achieved by sequential shotgun sequencing of a set of overlapping large insert clones. Good day, this is Professor Will, CBMD PhD. Today is Monday, August 26, 2019. It is 1:07:58 seconds Eastern Standard Time. And this concludes my reading on DNA sequencing. Hope you enjoyed listening. Have a great day.